Dear student, today we will discuss modern synthetic theory of evolution. Now, modification of Darwinism is called Neo-Darwinism. The Neo-Darwinism was proposed by various scientists: Huxley, Spencer, Jordan, Gray, Haeckel, Weismann. They believe that the natural selection has accounted everything that involved in the evolution, and they believe that adaptations result from from the multiple forces, and natural selection is just one force out of them. The neo-Darwinism matches to modern synthetic theory in 19th century. So this was the concept of neo-Darwinism, that the natural selection which was given by the Darwinism was true, and the evolution occurred by natural selection is also true. But beside natural selection, there were other forces which were also acting. So neo-Darwinism was later on modified to modern synthetic theory, and there were many other concepts that were added to the evolution. Now, here, if you see, the modern synthetic theory is based on Darwin's theory, Devary's theory of mutation, recent knowledge in cell biology, molecular biology, and genetics. So, it is stated that variation in a gene pool of the population occurred due to natural selection and genetic variability. So, natural selection and genetic variability both are important, and this population, it tells the unit of evolution is not individual but a population. So evolution is not acting on a particular species, on a particular individual, but is acting on the population in a particular area. This is it is a gene pool of the population that changes, evolve, and diversify. So this is what is the statement for the theory of modern synthetic theory of evolution. Therefore, speciation is due to the gradual accumulation of small genetic changes. So basically, the concept of genetic was introduced everywhere in the modern synthetic theory. Now, you know that gene pool is the total genetic diversity found within a population or a species. A gene pool is a collection of different genes, both expressed and non-expressed. So, for example, genes of all the varieties of mango. So, there are five forms in which the evolution occurs according to the modern theory. One is natural selection, second is genetic variability, third is isolation of different population of a species, fourth is genetic adaptation, and fifth is speciation. So here the genetic variability may be because of the genetic drift, because of the gene flow or mutation, because of the recombination. So we'll be discussing all these in the synthetic theory. Now, variation and natural selection are two main forces of evolution. The variation are introduced by mutation, recombination, migration, and hybridization. The variations finally result into the adaptation of the most suitable trait. The adaptation occurs at genetic level, so they are inherited. They inherited when the variation act on differential population group of a particular species. The isolation act as a physical force that help in the accumulation of variation in different groups only. The different groups evolve to new species called speciation. Okay, so according to this modern synthetic theory, the variation get accumulated, and the reason for accumulation is isolation of a particular population of a species by some factor. Okay, so this is the main reason for evolving of the new species. Now, we'll also discuss the mutation theory, which is given by Hugo de Vries, and let's discuss the merits and demerits. Hugo de Vries is known as father of mutation, and what he said that evolution occurred due to mutation. So he proposed the mutation theory for the origin of new species And according to him, the new species arise from the pre-existing one in a single generation, all of a sudden by distinct and discontinuous heritable change. And he called this as mutation. Mutation are responsible for variation among the population on which the natural selection is operated. So this was his concept of evolution. Darwin called the changes as sport, whereas the Bateson called these changes as saltations, that is single step large mutation. Now, key points of mutation theory is mutation are sudden, large, distinct, discontinuous heritable change. They may appear in different member of the same population and may be useful or harmful. Mutations are subjected to natural selection. The useful are selected, favored by nature, and harmful or lethal mutations are eliminated by death of mutant. Evolution by mutation is discontinuous and jerky process. Mutation theory explain about the occurrence of vestigial and even the over specialized organ. For example, appearance of the short leg and corn sheep to normal long legged parent. So, what was the objection on the theory? It cannot explain coevolution. It cannot explain the frequency of mutation. It cannot explain the natural selection because the frequency of mutation is very, very low. Most of mutation are recessive and they cannot be expressed. So, its utility in the progeny is undetermined, because since if they are not expressed, how will you come to know that mutation has occurred and is responsible for variation? So the modern concept, so again the modern version of this theory is the modern concept of mutation, and it states that all the organisms are subjected to mutation. They occur in all these cells. Mutation in the germ cell are heritable. Mutation can be harmful, useful, needful, and could be recessive or dominant. So all kind of mutations are there. It occurs in all the cells, all the organism. Mutation form raw material for evolution, but alone cannot produce new species. So it is one of the factor which is responsible for variation, or we can say further the evolution. Now let's come to the population genetics and understand what is this. The 1930 pioneers were Fisher, Haldane, Sewall, Wright, and all those. And population genetics is a field of biology studies the genetic composition of biological population. So here we are not concerned with individual, we are concerning the gene pool, that is for the whole population, and their changes in the genetic composition of the population. So when we are considering a population, we are studying what all genetic changes are there in the population as a whole. Not, we are not studying a particular individual due to natural selection or mutation or any other force. It is helpful to study the pattern of genetic variation in population. The population genetics, um, when the natural selection is operated on population, some variant arose in population which may get adapted better, and due to these changes genetic makeup of the population changes. So whenever there is environmental change, then what will happen, there are variant which are already there in the population and they adapt this change very well. This is what is the theory of natural selection. Okay, and when they adapted means their genetic makeup changes. So over a period of time, because of the differential reproduction, the population, the genetic makeup of the population changes. Okay, so Mendelian genetics is a specific genotype frequency produced with the controlled crosses. Population genetic is the frequency produced in the entire population. So this is just to differentiate how the population differs from the Mendelian genetics, population genetics. So it depend on the population genetics, depend on the mating system, natural selection, mutation, random chance, all these things. Now, Mendelian population is an interbreeding group of organisms that share common gene pool. All members interbreed freely, that is free gene flow, and show random mating. So in a population there is random mating, so free the gene flow is there. Now, what is gene flow? The movement of gene from one population to the other population within the same species. We are talking about the same species. For example, monkey from North India breed with a monkey of West India. Okay, so if you consider this is population 1, okay, of any animal, say monkey, they are migrated to the new area. So there already exists some population 2. Okay, now this population is receiving population number 1. The migration introduced the alteration in the gene pool. So rad 1 is having their own gene pool. Okay, sorry, the gene pool was there of combination of both uh, 1 and 2, but when the one come near to 2, then there is hybridization, of course crossbreeding, so that there will be genetic variability. So the gene population, the gene pool of the population changes, which leads to the genetic variability within the population. Therefore gene flow is important source of genetics. So whenever there is gene flow, or we can say there is migration of population from one to the another, then there is always a genetic variability that get introduced. Okay, so this is one reason for introducing the genetic variability or variation, we can say. Now next we'll talk about the gene frequency or allele frequency. It is the ratio of specific allele in the population to the total number of allele at the same time at the genetic locus. It is expressed in the percentage. For example, there are two alleles for the height of plant, tall and short. In homozygous condition of population for tall plant, all the allele would be T, that is capital T. So frequency of T would be 100%. If all T are there, definitely it will be, or if all small t are there, again frequency will be 100%. But if half capital T, that is the tall, and half short, they are there in the genotype, in the allele form, then we'll say there is 50% of inheritance to the next generation. So 50% frequency is their allelic frequency, and it is calculated as frequency of one of the allele is equal to number of allele of that particular allele in the population upon the total number of both the alleles in the population, or as many alleles in the population. Allele frequency is the most commonly calculated using Hardy-Weinberg equation, which describes the relationship between two allele with the population. So next video will be discussing about the Hardy-Weinberg principle. Thank you.